Hi, and welcome to this video. We're going to be running through some multiple choice questions provided by the OCR exam board on photosynthesis, so that's module 5. So what you should do, if you haven't got the questions, is you follow the link in the description, go to the questions, skip past all the answers, do them, and then come back here to sort of see how you work out and get to the answers for each one. Okay, let's go. So question 1, which two reactions happen during photophosphorylation? So this is the light-dependent part of photosynthesis, the sort of first step. ATP is hydrolyzed, no, that's wrong. NADP is reduced, that's correct. ATP is hydrolyzed, no, wrong. NADP oxidized, that's wrong. ATP is synthesized, yes, that's correct. NADP is reduced, that is correct. So therefore this one is, yeah. So the answer is C. Moving on. Now, this question is pretty mean. It's kind of a trick in a way, so I'm zooming in a little bit. Graphs 1 to 4 relate to photosynthesis. They show the absorption spectra of different photosynthetic pigments, with light absorbance on each y-axis. Now, this is the really important bit. One is the action spectrum of a plant containing these three pigments. So the action spectrum is the total photosynthetic output across different wavelengths of light. So it's a kind of combination of all the pigments together. So that's the first thing that we should really start at. Which one looks like a combination of all the other kind of graphs added together? And it is this one here. Okay, so this one is kind of the, if you look at the area under this graph, it's like you've added all the graphs together. So this one is the action spectrum. 2, which basically takes us straight to the answer there. It's that one. The action spectrum is 2. We can look at the other things and just check that they make sense. Chlorophyll A is going to be this one. So yes, that's correct. It could have also been that one. Chlorophyll B is going to be number 4. And this would be carotenoids. So yeah, it is B. But it's a tricky one there. I have never seen that sort of question before. But yeah, approaching it via the action spectrum idea is the best way. If you need to research that, I'd write down the definition for action spectrum, Google it, and write it down. 3. 2 plants with variegated leaves were supplied with air containing radioactive carbon dioxide. After 24 hours, a leaf from plant X, which had been kept in the dark, and a leaf from plant Y, which had been kept in the light, were compared by measuring the radioactivity in the leaves. The results are shown in the diagram. Okay, so let's just highlight this one. This is the light one. So obviously we can see there's a lot more radioactivity in the light leaf. What explains the radioactivity measurements in the yellow zone of leaf Y? Okay, the yellow zone is here of leaf Y, so we're talking about this measurement up here. Okay, let's just look at the answers. Carbon dioxide is fixed and stored as radioactive GP in the yellow zone. Well, I'm kind of questioning this one because there's no photosynthesis happening in the yellow zone, but potentially there could be some ATP to power the light independent stages in the yellow zone, but I'm not really sure. So I'm going to keep moving on. B, fixation of radioactive carbon dioxide occurs more slowly in the green zone than the yellow zone. No, that's definitely not true. Green zone would be much faster. C, products of the light independent reaction move from the green zone to the yellow zone. That could be true. So products of the light independent, so e.g. glucose, right? So we could have radioactive glucose made in the green zone, spreads across. So that could be true. That could well be true. I'm going to put a question mark next to that one. D, radioactive carbon dioxide diffuses into the yellow zone. No, and accumulates here. So C or A? I think C is the best answer here. Yeah, C. Because there's definitely going to need to be some glucose going into here, otherwise the cells will die. So yeah, definitely C. Moving on. 4. The diagram shows a chloroplast. Which line in the table shows the location of processes occurring within a chloroplast? Okay, photophosphorylation is going to be occurring in the thylakoid, so that's K. Regeneration of RUBP, that's going to be occurring in the stroma, so that's L. And transcription is going to be occurring at M because that's the DNA within the chloroplast. So yeah, C. Okay, moving on to 5. Which reaction is catalyzed by the enzyme Rubisco? So, well, kind of tells us actually the name of the enzyme, ribulose bisphosphate carboxylase. So it's carboxylation of ribulose bisphosphate. That's just A. Okay, moving on. 6. Which pair of areas within the chloroplast will show the steepest pH gradient between them? This is between the place where the hydrogen ions are concentrated and the place where they're taken away from. So no and maybe, but no and maybe. Maybe and, well, yes and not really. This is the one. Yeah, D. Stroma and the thylakoid space within the thylakoid membrane. I forget exactly, it's not in your syllabus what the gradient is. I think it's 3 full pH units, which is a lot. 3 full pH units. If you do chemistry, you'll know that that is not 10 times, not 100 times, but 1,000 times gradient between the two. Okay, 7. Here is some TLC apparatus, thin layer chromatography, investigating photosynthetic pigments. As the solvent rises up the plate, the pigments are carried up and separated, producing a vertical line of coloured spots. So we get sort of, blobs like this. Different colours are, I can't be bothered to do different colours, but yes, we get blobs, slightly different colours like this going up the top. What distances need to be measured to prove via the calculated RF values whether the pigments in each leaf are different and the same? From the spot at leaf 1 extract to the midpoint of each coloured spot. I'm not very good at this one. From the pencil line to the top of each coloured spot and to the solvent front. Yes, that's the one. Thank you. It's definitely going to be B, because we have to have 2 distances measured. We need to measure to the spot and to the solvent front. So there's actually something that I've learned, because I can never quite remember whether you're supposed to remember to the middle or to the top. So here it is from the exam board. You measure to the top of each coloured spot and the solvent front. It's definitely not the bottom. That's definitely not that one. So the answer is B, 7B. So, yeah. Moving on. Blank page there for some reason. Okay, question 8. Sampling a suspension of isolated chloroplasts at intervals allows the measurement of concentrations of photosynthetic intermediates to take place. Measurements are taken before and after removing the source of carbon dioxide to the chloroplast. Okay, so we have carbon dioxide and then we remove it. So that's, sometimes you have to think what's going on. Before and after removing the source of carbon dioxide. So we have CO2, then we remove it. Which line of the table shows how the concentrations of ribulose bisphosphate and triose phosphate were expected to change? Okay, so you need to think about, the Calvin cycle. CO2 is kind of coming in here. You've got RUBP just before the CO2 comes in, and then you make GP, and then you make TP. Okay, so it's kind of like this. So if you suddenly remove CO2, the concentration of RUBP is going to build up. So it's going to increase because it's like a traffic jam. It's like a roundabout or a roundabout. So if there's an accident at a roundabout, the cars would build up behind the accident. So then we get a build up of RUBP, and GP and TP would decrease. So that increases and this one decreases. So therefore the answer is B. Moving on. Which statement correctly outlines some of the main events in photosynthesis? Okay. A 5-carbon carbohydrate accepts carbon dioxide, that's correct, to form temporary 6-carbon intermediate then splits, and is then reduced by an ADPH derived from photophosphorylation. Yes, that's true. That's all true. So I think it's probably going to be A, but let's just check the other ones. B, a 3-carbon carbohydrate is regenerated and reduced by hydrogen molecules derived from. No, that's wrong. That bit. If that was 5C, it would be okay. Photolysis uses light to produce reduced NAD, yeah, and oxygen. No, oxygen is not used, so that's the why that bit's wrong. Photolysis produces NADPH, yes, and ATP, which I'll use to reduce a 5C carbohydrate. No, that's wrong. So the best one is, yeah, 9A. Tricky one, that one. 10. Okay. Oxygen use. Good coffee. Oxygen use and release by a plant was monitored for 12 hours of darkness and 12 hours of bright light. Temperature and all other variables kept constant. The results are shown in the graph. Calculate the volume of oxygen used by the plant in respiration over the duration of the experiment. Okay, so this is all about, it's one of those like attention to detail questions. They love this sort of question. They kind of like really make you just read the question and really understand what they're asking you. The thing it's asked you here, used by the plant in respiration. Used by the plant in respiration. So how much oxygen does the plant use in respiration? Where can we find the information in the graph that is just respiration? Well, it's here, 50, okay? When it's dark, the plant is using 50 cm cubed per hour of oxygen. That's just from respiration. There's no photosynthesis occurring. So how much would be used just from respiration over the whole 24 hours? Well, it's 50 times 24, because there's 24 hours in a day. Cm cubed per hour. What's 50 times 24? Hang on a minute. 1,200 cm cubed. The answer is D. Okay, by the way, that right there, light compensation point. Okay, moving on. 11. Which chemicals are both the products of respiration and the raw materials of photosynthesis? CO2, for sure, and products of respiration, no, yes, no, no, yeah, B. 12. What name is given to assembly of several hundred accessory pigment molecules around a molecule of chlorophyll A? I think 12C photosystem. Yeah, that's right. I paused there for a moment because sometimes at university level you might call that the antenna complex, because I think the photosystem is more technically is the protein as well as the pigment molecules, whereas I think the antenna complex is maybe just the pigment molecules within the protein itself. But anyway, photosystem is the best answer. 13. The graph shows the interaction of two factors that affect the rate of photosynthesis. Carbon dioxide and light, basically. Now, if we look at that, we've got CO2 down here, and we've got low light and high light. So those are the two factors. Which of the following statements correctly describes the relationship shown on the graph? Okay, so at high light intensity, an increase in carbon dioxide concentration from 0 to low causes an increase in the rate of photosynthesis. No. That would be if we had light intensity on the x-axis and then the two lines were labelled with CO2. At high light intensity, again, no. At low carbon, at high light intensity, an increase in carbon dioxide concentrations from high to very high. Okay, at high light intensity, an increase in carbon dioxide from high to very high does not increase the rate of photosynthesis. Yes, I think that's true. So that's basically saying that if we're here and we move to here, there is no change. Oh, hang on a minute. Sorry, I was too quick to discount this one. At high light intensity, an increase in carbon dioxide concentration from 0 to low. So yes, that would be talking about from here to low. Okay, it does increase. So we're moving, this is the first one. This is statement 1, so that's true. Apologies. Statement 2 is also true. What about statement 3? At low carbon dioxide concentration, an increase in light intensity from low to high does not change the rate of photosynthesis. I think that's false, because that's kind of like sort of down here. So if we can move it, it's talking about, I'm going to do this in blue so we can see it's moving from there up to there. It does increase, not by a huge amount. So I think that one's false. So I think only 1 and 2 is the correct answer. So 13B. Am I right? Yes, that's the right answer. Okay, moving on. Ooh, this looks horrible. Gosh, right. Algae grown in a culture supplied with oxygen gas and water, H2O, containing the oxygen-16 isotope. I guess both of them have? Yeah, okay, so oxygen-16 isotope. The algae were then briefly supplied with oxygen gas containing a mixture of the oxygen, oh my gosh, oxygen-16 isotope and the oxygen-18 isotope. Over the course of the next hour, lighting conditions were varied and the concentration of the two isotopes in the culture were measured. Oh my word, this is horrendous. I'm just going to color code this so I can kind of think clearly. I kind of think in colors. Okay, right. Which of the following statements explain the trends seen in the data? Well, first let's look at the data and try and figure out what's going on. Okay, so in the dark. Okay, so well, this is what I think we've got to look at. The water has got the oxygen-16. And then when we've got the gas has more 18. Right, okay, so basically what's happening is when we've got light, goodness me, this is really weird. So when we've got light here and here, what is happening? Water is being split into oxygen. So the oxygen that's in the water is only the oxygen-16 variety. So therefore, that's what's going on here. Oxygen-16 is being produced, and over here, oxygen-16 is being produced by the light. The level of oxygen-18 is being used up, I think, because of respiration. So the oxygen gas is being used up by respiration at a steady rate. So okay, this is falling. Respiration, and this one kind of, well, it increases photolysis. Okay, so let's look at the statements. Hopefully that's right. Okay, so statement 1. The concentration of 18O2 decreases at a constant rate irrespective of light or dark. Yes, due to anaerobic respiration. That's wrong. A little distractor there. It should be aerobic respiration. Statement 2. The concentrations of both isotopes decrease at an equal rate in the dark due to both molecules fitting the active site of the last enzyme in the electron transport chain. Well, yes. Yes, that's true, because both oxygen-16 and oxygen-18 can be the final electron acceptor in respiration. Oh. Statement 3, the concentration of 16O2 increases in light due to photolysis of water. Yes, that's right. Wait, hang on a second. Photolysis of water, that was previously produced by oxidative phosphorylation. I think it's 2 and 3. I almost thought it was only 2, but there's no option, so it's got to be 2 and 3. So the answer is C. Am I right on that one? Which question are we on? 14. Yeah. Gosh, that was a horrendous question. Well done if you got that right. I really had to think about that one. Okay, question 15 here, and I have to confess I got this one wrong. First time I tried to do it, and I think one of the things is, like, if you have to concentrate so hard. On the previous question, 14 required a lot of concentration. Then you get to 15, you're just like, oh, this is easy, just quickly do it. You must keep your concentration the whole way through. This is about, like, sort of your concentration kind of budget in your brain. You keep that focus the whole way through. 15. Okay, so I made a mistake here because I just went for, immediately, I went for, like, okay, starch 5/6, and, sorry, starch 1,6, and ribulose 5,6. But I didn't read the question carefully enough. It says to give maximum sustainable growth in a plant. Now, if we want the plant to grow, it needs to be making proteins. Starch is really a store of energy, so we want to actually have 5,6 for ribulose bisphosphate. That's always going to be that way, but we'd like to divert the other 6 into proteins. So the answer is A for that one. All right, moving on. Ooh, okay, let's zoom out a little. The graph shows the results of an experiment using a culture of illuminated photosynthesizing algae. After 300 seconds, the carbon dioxide supply was stopped. The concentration of glycerate 3-phosphate and ribulose bisphosphate were measured over a 400-second time period. Okay, so that's basically, we've got light, and then the the carbon dioxide supply was stopped. That's what's happening. So then we're looking at G3P and RUBP. Again, let's think about the Calvin cycle. We've got Calvin cycle like that. We've got CO2 coming in up here. We've got RUBP. We've got GP. We've got TP. Okay. So when we remove CO2, we'd expect an increase in RUBP, which is what happens, and GP would come down. Which statement explain the changes between 300 and 400 seconds? GP is converted into RUBP. Yes, that is true. Not directly, but it is. It goes from here round to TP, then to RUBP. RUBP is converted into GP. No, that's not going to be occurring. RUBP is not fixing carbon. Ooh. I wouldn't say that RUBP is not an enzyme, so I wouldn't say it's fixing carbon. Is there an option of only one? Or is there an option of one and there isn't an option of 1 and 3, so it must be only one. The reason this is not true is because RUBP is not the enzyme. RUBP is a 5-carbon molecule. If it was ribulose bisphosphate is not fixing carbon, that would have been correct. So the answer is 16D. I had to think about that for a second because I just checked the answers from the exam board. I'm pretty sure they've made a mistake there because the exam board says 16C, but I'm almost certain that is true. Sorry, that is false. D is the right answer. It definitely can't be C because 3 is wrong and 2 is also wrong. Statement 2 and 3 are both wrong, so it's definitely not C. I think the exam board made a mistake there. Answer is D. Moving on. 17. In an experiment to measure the rate of photosynthesis of a water plant, bubbles of oxygen produced by the cut end of a stem were collected under a funnel-shaped end of a glass tube, like this. Bubbles, 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 up there. After 20 minutes, a syringe attached to the far end of the tube was used to draw the oxygen ball up the tube so that length could be measured in a millimetre scale. The mean length of the bubble was 12 millimetres in a tube diameter of 0.5. Okay, there's loads of data here that we've got to pick out. Okay, first of all, 20 minutes. Second of all, 12 millimetres. Third of all, 0.5 millimetres. So what have we got to do? Well, we've got to use pi r squared L. So pi times r. The r is not 0.5 because that's the diameter, so it's 0.25 squared times 12 is the millimetres, but we want this per hour, so what would it be if it was per hour? This is 20 minutes, so we want to times it by 3, because 20 minutes times 3 is an hour, so we have to times it by 3. Okay, so let's do that. Turn my phone sideways to get pi. Where's pi now? Pi times 0.25 equals times 0.25 equals times 12 equals times 3 equals 7.06. So the answer is A, 7.1. 18. In an experiment to investigate the effect of wavelength of light on photosynthesis, the student added blue DC-PIP solution to a suspension of isolated chloroplast. DC-PIP turns from blue to colourless when it accepts hydrogen ions and electrons. The DC-PIP chloroplast mixture was kept in the dark until needed. then a sample is drawn into a capillary tube and illuminated by coloured light of a known wavelength. Which statement about the expected results of the experiment are correct? DC-PIP will decolorize at different rates in different wavelengths of light. That is correct, because DC-PIP is kind of is acting like NADP, so it can be reduced. And when it gets reduced, when it has hydrogens added, it goes clear. So different wavelengths of light will make the light-dependent reaction of photosynthesis happen at different rates. So one is true. DC-PIP will decolorize faster in red than in green. Yes, that's true. The end point of the reaction will be a colourless solution. I think that's true as well. So I think 18, 1, 2, and 3. Oh no, it says B. I got that one wrong. Apparently it's B. I guess because what is the end point of the reaction? I guess. Is there an end point of the reaction? I mean, I guess it's, yeah, I mean, that's a pretty hard one. The answer is B. I think the exam board is saying that even though the DC-PIP itself will ultimately be fully reduced, it's not really the end point of the reaction from the photosynthesis kind of angle. You know, the photosystems are still going to be churning out reduced NADP. They're still going to be splitting water, so the reaction doesn't end. Even if all the DC-PIP has been decolorized, I guess the reaction hasn't ended yet, or never ends really. 19. Corals are colonies of marine animals that contain photosynthetic algae inside their cells. Environmental changes can result in coral bleaching, where the pigments in algae are lost from the coral. This is a horrible thing, coral bleaching. Look it up, it's very, very worrying. Which statements support the idea that algal photosynthesis is important to corals? Bleached corals grow more slowly. Yes, that would support the idea. Corals receive carbohydrates from the algae they host. Yes, that would support the idea as well. Carbon dioxide from coral respiration is used by the algae in their cells. I would say no to that one, because that kind of supports the idea that coral respiration is important to the algae, not the other way around. So I think the answer is 1 and 2, 19B. Yes, that's right. 20. Farmers can increase the yield of raspberries by growing the plants in plastic tunnels fitted with irrigation systems. Which statement explains the improved yield of raspberries in a plastic tunnel? Okay, well, we want to think about things that increase the rate of photosynthesis. So I don't think reducing transpiration affects photosynthesis. I don't think. Higher temperature in tunnels increases the rate of the light-dependent reaction. Yeah, higher temperature, yes, that could be true. And 3 as well, yeah, that also could be true. So I think 2 and 3 only will increase the yield of raspberries, not 1, because it's not about photosynthesis or fruit. So therefore, 20C, yeah, that's correct. All right, so I hope you found that useful. You can look at the ones you got wrong and sort of focus your revision accordingly, and I'll see you next time. Thanks.